Hello friends, welcome to TECHQUEST channel. This is for the medical lab technologist by EarTech. Some of you have a question about the calculations in manual cell count using Newer counting chamber. This video explains the calculation. The cell count is done on a body fluid that may be extracellular fluids like blood, transcellular fluids such as cerebrospinal fluid, synovial fluid. This provides lubrication and cushioning for organs and joints. The cell counts are reported per microliter, that is, number of cells present in defined volume of sample. The number of cells present in 1 microliter is equal to 1 cubic millimeter. The amount of cells present in the area of 1 millimeter cube, that is 1 millimeter length, 1 millimeter width and 1 millimeter height cube, just like a measuring cup having 1 millimeter into 1 millimeter bottom and 1 millimeter of height. To make it a uniform unit, all cell counts are reported in cubic millimeter or 1 microliter. Both are same. But the number of RBCs are in millions, the WBCs are thousands, platelets are in lakhs, and other body fluids like CSF are below 10 in normal conditions. Using a same counting chamber for counting few to some millions are made possible by making dilutions and the counting areas adjustments. The calculations are made with the dilution factors and the area counted for each type of cell count. Let's calculate from the Newer counting chamber. The counting chamber has 9 large squares, each measuring 1 millimeter by 1 millimeter. But the 4 corner areas are subdivided into 16 small squares for counting WBCs. The center square is divided into 25 squares, and these 25 small squares are again divided into 16 very small squares. These are used for RBC count. Since the RBC count is very high, the area counting is less. These small divisions for each 1 square millimeter helps to count the cells accurately, avoiding the repeat count of each cell. Also, it gives the idea of proper dilution, mixing and proper charging of the sample onto the chamber. When we calculate for 1 square millimeter of area, that is the area counted. Depth factor. If you watch carefully on the counting chamber, there are 2 elevated mounting supports on both sides of the counting area. When you place the cover slip on the counting area with these mounting supports, you will notice that there is a gap between the counting chamber counting area and the cover slip. This is the area we are going to load or charge a sample. The gap between the cover slip and the counting chamber is 0.1 millimeter. Printed in each counting chamber. Different depth counting chambers are available for research purpose. So for calculation of cubic millimeter, the height is not 1 millimeter, but it's 0.1 millimeter. So when we calculate for cubic millimeter, we need to multiply into 10. The reason for counting 1/10 of the millimeter is to make the cells visible in thin single layer. That's the reason we keep the charged chamber in a petri dish for few minutes to settle so that we avoid the recounting of the same cells. The depth factor is stable as 10. For counting chamber, we use thick cover slip. The thin cover slips used for urine microscopy cannot be used as these cover slips easily bend and make the depth inaccurate. Counting area. We are reporting the cell count for cubic millimeter. So whatever area we count, we need to calculate for 1 square millimeter. Usually for WBC count, all 4 corner squares are counted and divided by 4 to get 1 square millimeter. Counting 4 squares and taking average gives more accuracy. For RBC count, the center RBC square is counted. Usually 4 corners, small squares, and 1 middle square is counted. Since the amount of RBCs are more, These 5 small squares represents the uniformity of the count. We are counting only 5 small squares in 1 millimeter. The area counted is multiplied by 5 to get the count for 1 millimeter. That is 25 small squares make 1 millimeter. We are counting only 5 squares, that is 5 by 25 equal to 1 by 5 square millimeter. For platelet count, we count 1 WBC square, that is 1 square millimeter, so that counting area is 1. So there is no multiplication for platelet count. For any body fluid, the area counted is taken into account, and if less than 1 millimeter is counted, the area is calculated for 1 millimeter by multiplication. If more than 1 square millimeter is counted, the value is divided to bring it to 1 square millimeter. Sample dilution. The sample dilutions are made according to the type of cells with their normal range. The dilution is essential to disperse the cells in a thin layer and avoid overlapping of the cells. Normally, for RBC count, 1 in 200. For WBCs, it's 1 in 20, and platelets 1 in 100 is made. For body fluids, the dilutions can be made with the amount of cells present in the sample. CSF, like body fluids, normally do not have many cells. These can be counted directly without dilutions. Diluting fluid. The diluting fluids preserves the cell osmotic pressure to maintain cell integrity to count. WBC counting fluid may have 1% acetic acid to lyse the RBCs because the WBCs are our interest. During calculations, the dilution factor is calculated for the amount of sample added to the chamber. For calculation, for any fluid and any cell count, the calculation is number of cells counted multiplied by depth factor multiplied by dilution factor divided by area. The number of cells counted at the particular area, the depth is constant. The dilution is made according to the cells of interest and number of cells. For CSF, it is normally undiluted. So the dilution is 1. Area counted differs according to the cell count. For CSF, to make the calculations easy and the number of cells are low, it is better to count all 9 square millimeter and any one of the square millimeter again, so that the number of cells counted multiplied by 10, that is the depth factor, multiplied by 1, that's the dilution factor, divided by area counted. Here we count 10 square millimeter. So the depth factor on the top, area counted on the bottom gets deleted. The number of cells counted is directly reported, the cells present per cubic millimeter. Thank you. Thanks for watching.